that compression of the air will be converted in the air into sending a signal of electrical conduction of spikes in a particular way. And these spikes, the frequency or the amplitude or whatever appearing in these forms of spikes will contain the information of the sound of the pressure. Hi, Sirago. Okay. Into the air, and this form of coding, this form of spikes, will be moved along the auditory nerve to the central nervous system, and will elaborate information. What signal, what's background, how can I enhance the signal to background ratio in order to get as much properties as possible of this kind of information, and disregards what is just rubbish. Okay, so in this kind of elaboration of the information, we will take the information that is good for our life and then disregard the rest, and then we will represent inside a particular structure in the central nervous system what is the pattern of the general organization of the world. That is the representation. So this will be the next topic. How can we represent several properties of this world into particular structures in the brain? And finally, we will find cognition, attention, motivation, and working memory. That is, whenever we are managing this information with already represented information, with stored information, retrieve this information, and then start to elaborate a response. This response will be followed by the next curve that will be from perception to action. Okay? Okay, our working plan here will be that we will open, as in last course, we will open each week a topic. Okay, the basic rule here, I'm continuing saying, I'm not going to extend to the lay closing. So the topic will finish by Sunday next week. So in order to start on Monday the next topic. Okay? I'm not sorry if you are just passing a bad issue, because sometimes there are unpredictable. Maybe you can suffer for something. Okay, some you can become sick by the end of the week or something like that. So in that case, my advice is to start as soon as possible each topic. Okay, if we are having the presentation on Monday or Tuesday, you are having this information fresh, so you can read it, you can redo, you can, and then by Wednesday, at last, you can finish all recordings and all activities. So you can finish it, and you can become relieved of being done a work, and you can get as much grade as possible, so that you are not afraid of having any other trouble. Okay, I'm not going to extend the deadlines. Okay, you're having the documents, you're having the presentation, you're having the activity and auto-evaluation, and then the topic will be closed. Okay. The calendar. Okay, so the basic calendar, so we are going to start. Today we are starting with this, and then we will open each week, and then but we are going to finish by 15 October, so that we can fix 22nd of October as the day for the exam. And the day of the exam will be similar. Similar is not identical. Will be similar to questions from each topic, from each auto-evaluation, but will be also some new new questions that will reflect how much have you taken and how you afford this information in order to solve aspects that have not been included directly in the course. But you need the course to solve these issues. So you can get 7 over 10 or 14 over 20 to if you are followed every for the 3-4 questions, the special questions, you need to get more data. You need to manage data from several staff, combine it in order to get a response. Okay. Okay. And best luck. Okay. Best luck to finish all this stuff. Okay. So we can get as much. Hopefully, we can. Everybody can get a maximum grade, and they can manage everything. Another thing. Well, okay. So bye bye from now. Okay. This is. I'm not going to move away. We're going to have first talk. It's just a general indication of how to do. We are also having the recording from last year, so you can work already with as presentation. Good in the upload in the system. Okay, this is the first topic. That is the transduction of sensory signal. Okay, so first, what is integrative neuroscience? This is the first topic. I need to start with this because or this integrative neuroscience, neuroscience of system, is some kind of science in which we can analyze as a general way information coming from different sources, combining it into a general background, and then elaborate a response. So, integrative neuroscience deals with information coming from outside, but also information coming from outside, inside our body, okay? Which is our internal state. It is okay, it is having some trouble. If it's having some trouble, it means that we can have some pain, for example. A function, the liver, is not working well. And then I need to know that there is something that is wrong there. Okay? Or I'm having a cut on the skin and this hurts, so I need to be careful with this cut. And then I need to keep it clean and all this stuff in order to recover from this disease. Okay? Okay, so the issue is just once we are having the external and internal information, how can we integrate these words and perceive it as a whole? Okay? So this is managed inside the brain. This is a particular issue that happens in several parts of the brain. And what we can do is figure out how this flow of information between this part of the world is driving a response, and we can figure out how this network is working in order to elaborate a response. If we figure out how this network is working, we are giving properties to each node of the network and give a special in order to get a response. Then we can see if this network is behaving the same way as the network, in such a way our network is predicting the response that can be also predicted from the biological network. In the sense, in the level in which the computational network is predicting the response, there is a fitting between these two models, or the model is fitting with the reality. Okay. And finally, what we can see with all this stuff is see how our response can change the behavior. Okay? The behavior is the response to the processing of the external and internal information. Okay, in this course, we're going to follow the different steps. The first topic is transduction. That is the one that we're going to hear. Then there is a coding. That is once this information is translated into change in the electrical act, and then this electric activity is containing the information that is being processed. Maybe some kind of information can be lost. And not all the information of the properties from the sensor systems are arriving. For example, touching the liver is not perceived. It's only perceived pain. Okay? So the liver is conveying information regarding pain, but not regarding touching. Okay? Okay, then with the coded information, we need to compare it with several different parts of this coded information. We can elaborate this information and enhancing the basic properties that are needed to our survival, and disregards what is rubbish, what is not good. Okay? That's elaboration of the information. Then representation, that means that the external wall or the internal wall is represented in several parts of a particular neural organ. For example, in the cerebral cortex, everybody knows the homunculus, in which we are having represented the entire body over the presental cortex. Sorry, the post-central cortex. And then cognition, attention, working memory. Okay, then we are going to dig into particularly dig into transduction. What's transduction? Transduction is the first step, in the perception of a sensation. In this transduction, what we are making is converting an external stimulus, that is a form of energy that comes from inside, maybe the occurrence of a particular molecule in a concentration of a particular threshold, or a mechanical change of the milieu, or a particular wave, that may be a mechanical wave, something else. Okay, this form of energy over the external milieu is changed into the properties of the internal of the membrane of the transductor. The transductor is one of cells. Then it happens several signal transduction, several metabolic pathways inside the cells that are converted into a form of energy that makes changes in the concentration of the transmitter, so that it appears a change in the electrical properties of the cell outside the cell, a neural component that will be sent to the central nervous system. Okay, so we are having at the end an action potential. Well, a couple of action potentials. Okay, so this actions potential will code the information that are given from the outside world. Okay? So that, for example, in this case, the frequency of the action potential, that means how close are one action potential from the other in temporal. If we are having 100 action potentials per second, so it means Earth, or 200 or of 1 kilohertz will mean that we are having 1,000 action potentials per second. So it's 1 kilohertz, and this will be a code of the amount of sugar molecules that appear outside. So in this particular case, the concentration of the sugar will be out by the frequency of the action potentials. Okay? The more molecules, the higher frequency. Okay, we are having different kinds of receptors, that is, photoreceptors are physical receptors. Mechanoreceptors is whenever we're having a movement of the media in which the transductor is. Receptors are receptors that are sensitive to the occurrence of several compounds outside the cell. And nociceptors are a special kind of receptors. In fact, are receptors that are sensitive to damaged cells that are delivering into the external wall several compounds, are all inflammatory compounds that are signaling several pro-inflammatory systems. Okay? Inflammation, histamine, and so on. So this could be the transduction happening that is happening in. We are having. Oh, sorry. No, okay. I was thinking. I was. So this is the transduction that is happening in the retina. Okay? The particular case of the retina is that all along this pathway is being converting it's been done the transduction. So it's the flow of electrical activity all along this pathway is a special kind of transduction of the signal, and this only the ganglion cell one that codifies the information. So at the end, all the information regarding points of light by the side of points of darkness are codified at the last ganglion cell. So we are having 12 people. Very good. Good success. Okay. So all along this pathway, what we're having is just a conversion of this source of energy that are photonic energy into a form current. Okay? So the first part, the first step of the transduction is the photoreceptor. That is a compound that is the 11-cis-retinal, which is sensitive to the skin. In this bound, we are having this bound in cis way. They observe that the radical, that is the external part, in both cases are on the same part of this double bond. Okay? And what happens is that the light is able to break this bound and change the configuration and transform it into the 11 to all transretinal. Okay, all these double bonds are in the transway, that are the biggest part of this bound are on the opposite sides. This part, opposite of this part, all this part. Okay. So this change in this molecule makes a change in the part that is containing this molecule. And this part is a protein that is the opsin. Okay, and here we are having the retinal here, and this is the part that is in the middle of this opsin. And this opsin is a 7 transmembrane molecule. We are having here 1, 2, 3, 4, 5, 6, and 7. 7 transmembrane domains. And in the middle, we are having the opsin. So that when this compound is changing, the configuration also changes. Okay? And this opsin, this protein, makes the retinal sensitive to different waves. In this case, we are having the blue cone, we are having light and darkness rods, the green one, and the red one. Okay, so all these molecules differentiate in the proteinic part that makes the 11-cis-retinal sensitive to a particular wavelength. Okay, but what happens electrically into the rods and cones? If we attach an electrode inside the cell, we can record what is the movement of the charges at both sides of the membrane. And what we can see here is that whenever we're having a flashlight, what happens is proportional to the intensity of light, a decrease in the flash response. Okay, so it means that in principle, whenever the highest intensity of the light, the lower level of hyperpolarization. of the cell. Okay? So this cell will be strongly inhibited. Oh, sorry. Okay? What happens? This is the representation, so we are recording here. And what happens, to our surprise, is that this rod cell, whenever we are having illuminating, the cell becomes hyperpolarized. Why this neuron hyperpolarized? Because of a special structure that happens between the membrane of the Conan rod and the membrane of the dishes in the segment of the constant rods that are containing this part that is sensitive to the light. What happens here? What happens is that in normal conditions we are having channel in the membrane of the rods and cones. So whenever in normal condition this porus is open, and if this porus is open in regular states with light, with a dim light or a light just normal, it is open, and it comes open so that the sodium can enter into the cell. So if the sodium can enter into the cell, the internal part that usually is negative in respect to the outside the cell, so becomes a little bit depolarized. Okay? So rods cells are usually depolarized. What happens when light comes to the receptor area? What happens when the light are going to the receptor is that the light hits the G protein, and the G protein disaggregates between alpha, beta, and gamma, and alpha part becomes free and becomes activated. And whenever the free alpha unit becomes activated, it joins the GTPase. Okay? Sorry, the GTP. And this molecule is the phosphodiesterase. So whenever we are having the phosphodiesterase, the alpha unit, and the GDP, there is a transchange of this molecule, and then it joins, the phosphodiesterase becomes activated, and the activated phosphodiesterase breaks the GDP, the cyclic GDP, the CGMP, and then the porus collapse. So if the porus collapse, it becomes closed, and then the sodium cannot enter. And as there are some pumps that are shutting down, are leaving and export the sodium outside the cell, the internal part of the cell becomes hyperpolarized. Okay? So this is what happens. So, resuming, in normal conditions, we are having in the darkness, we are having secret GMP, that tip is open. Sodium can enter and then depolarize the cell. Okay? But if light is coming, phosphodiesterase becomes activated, it breaks secret GMP, it closes the channel, and then as sodium cannot enter, the cell becomes hyperpolarized. So whenever we are having light, the cell becomes inhibited. Good. So this is the rank of vision. What happens in the hair cells, in the auditory system? The way that we are transducing the signal is a little bit different. These are mechanoreceptors, and the mechanoreceptors are net at the tips of the cell that are attached to the cilia, and the cilia contains a pulse. Okay, these are the nets, and these are the cilia, and these are the porus. If the porus is open, the potassium can enter, and then the cell becomes depolarized. Okay? If the movement compresses the net, then the net closes the porus. It's just mechanical. And then if the porus becomes closed, then the cell becomes hyperpolarized. Just simple. Okay? Okay, this is the movement. We can get several adaptations as the net move along the cilia. Okay? Okay, this is another kind of mechanoreceptor. We are having here for example, a patient corpuscle, and then we are having several nets that goes to the membrane here. And then if there is a deflection, a movement of this net, then it comes open, and then the positive charges can enter and then generates a zinc current. Okay? If a zinc current is generating at this point, we are having a Rambier node inside the patient corpuscle, inside this high structure, and then the Rambier node can work as a source. So there is a current of zinc and source and zinc, and this is a movement, and this movement is moved along this pathway and then communicating along the axon. Source, and then the polarizing axon, and then it appears here an action potential. An action potential that comes with this action potential. Okay? And this is a continuity of source and source and sink. Source and sink. Okay? These are mechanoreceptors. These are a special kind of mechanoreceptors that are proprioceptors. The same way, we are having a cell here that is sensitive, and then we are having the synapse here, okay, with the spindle afferent, okay, and then this movement, this network here, is what opens the porus, the channels, then allowing the positive ions to enter, and then it becomes negative and so on. Okay, a special kind of transducer are receptor in a special part of the cell. That's what is converting this action potential we're having here, this bipolar olfactory epithelial cell here, and in here we are having the change of activity. In this case is hyperpolarization also, and it's because there are some compounds here that touch with a special kind of receptors that are adapted chemically, conformationally. Okay, these are the distribution of the receptor. In this case, the olfactory epithelia are also appearing at the top of the membrane, in the olfactory mucosa, and these are the distribution of the cells. Okay, in this case also, in most chemical transducers, we are also having also G protein transducers that are receptors that is having the 7 transmembranes, and we're having an of this membrane protein to the alpha, beta, gamma G protein, components of the G protein. Then there is the delivery of alpha unit, and then there is activation and then opening of several channels that are transducing the signal into a change in the electrical properties of the cell. Okay, in that case, that are the chemoreceptors, we are having a variable part, that is these parts that are the outside part of the transmembrane subunits, and these parts are containing different conformational segments, so that we can get different parts, even to several, in this case, 2,000 kinds of genes encoding olfactory receptors. So we are having the possibility to differentiate between 2,000 kinds of molecules with several combinations. Okay, but we're having more or less one receptor to one compound, or one class of compounds. Taste is just the same. Yes, we're having over the porus, just like chemical interoception. For the olfactory, we're having vapors, just the gas, volatile compounds, and for this we are having compounds in solution. Okay. But at the end, what we're having here is just a final change in the electrical properties of this mimic here, and then the delivery of the delivery. This is the electrical change, and then there is the delivery of the neurotransmitter. Then on the postsynaptic cell, we are having transduction, the codification of the signal. We are having several kinds of chemical transducers that are perceived as mechanical. In this case, the vanilloid receptors that are chili and jalapenos and so on, that are spicy food, are chemical compounds, but at the end are moved as burning, and then are perceived as some kind of temperature receptors. So vanilloid receptors are receptors that is having properties to several ranks of temperatures, but also are sensitive to several compounds like capsaicin. So capsaicin are perceived. Sorry, where is that one? Is capsaicin is perceived high temperatures. That is, whenever you're feeling like spicy food. And this is what is transduction. Okay? And we will see you next Monday. Okay? Okay. Good. We have finished. Okay. Do you have any question regarding that, or regarding the course? Okay, go ahead, Ikram. Go ahead, Ikram. Okay, you're typing. A special kind of the terpenes is giving some colorful aspects to jalapenos and special spicy food. But in regular way, the vanilloid receptors that appears by the skin, in the skin, are sensitive to different temperature ranks, depending on the transmembrane. Receptors are sensitive to a rank of temperature higher, lower, and so on. And this is the way we are perceiving temperatures, the number of vanilloid receptors that are being activated in a particular time. Depending on this rank, we are having this. But in addition, whenever we are having too much receptors of this kind that are being activated, it is perceived as pain. Okay? Okay. So we will see you next next Monday, please let me know if you're having any concern along this week by reading, and if something is not well explained or so on, you can send me an email and I can find time to respond to you. Okay? Okay. So, bye-bye. See you next week. Bye.